All right. Can everybody hear me okay?
Yes, sir. Loud and clear.
Awesome. Thank you. Cool. Yeah. We're on to our last unit. I can't even believe it. We're in week 7. Got 2 weeks left, basically. And summer goes by so fast, doesn't it? I'm sure not fast enough for some of you. When is this ever going to end? But nonetheless, we are almost done. As such, we only have a couple of things left to go through. We have urinary system and we have reproductive system. That's it. That's all. I'm just slowing down here because do you think there's probably a reason for the urinary system to be saved until last? Just kind of waiting for an answer. I hope you guys realize there's a reason, right? I've been trying to say this all semester long, but physiology is comprehensive. And the urinary system is the most complex of all of the systems because it utilizes all the rest of these organ systems that we've been learning about. Even though I'm a cardio guy, I tend to end up saying that if somebody were to ask me what's the most important system of the human body, what runs everything else, oddly enough, I don't tend to lean towards the heart. I tend to lean towards that urinary system, and it's such a critical component of the human body. So I want somebody, anybody, everybody in chat, I want you guys to write down at least one function, don't press enter yet, at least one function of the renal system. Okay, write it down in chat, just wait. I'll tell you when to press enter. Write as many things as you can. Don't press enter yet. Oops. So go ahead, you guys can write one thing, you can write multiple things. What's a function, many functions? What are some of the functions of the kidney? Well, the entire urinary system. All right, go ahead and press enter. I want to see what you guys got. Ooh, I love filtration. Some of you guys have been actually doing some of your research beforehand. I love it. I love it. I got filtration, excretion, regulate ion levels. Great, Sabrina there. Filtration is its primary role, Andy. I love that. Tariq, you got your filtration in there too, although fictoration, I love that. Yep, it likes to fictorize things for sure. Get rid of waste, water balance, love it, eliminate waste, blood pressure, pH. Thank you, Ashley, for writing pH. Yeah, it's gonna control 30% of all the body's pH. Excrete waste, micturition. Ruju, you got down to the bladder. Thank you for that. And filtration. Good. Okay, these are all great answers. You guys should, screenshot this if you want to. Just try to get a hang on what everybody else is thinking. Now, if you were to talk to like the layman about the function of the kidneys, most people are going to say it helps you make pee, right? That's kind of the thought process of the kidney and the urinary system. Helps you make pee. And a lot of you guys, wrote in, get rid of waste, excrete waste, that sort of stuff. Now, I just want to make sure that it's known straight up that that is not even remotely the primary role of the urinary system. It's just a byproduct. Its primary role is filtration. It filters your blood. We've talked about this through the semester. 180 liters of blood per day. Just mind-blowing. 20% of your blood volume is going to go through your kidneys in the next minute. Again, staggering. 20% of that volume is going to get filtered. That's how much filtration is occurring inside of your kidneys. I hope that's making some impact in your brain. I mean, that's 180 liters is 92-liter Coke bottles side by side. You've gone to the restaurant and seen those 2-liter bottles side by side. You're probably not seeing 90 of them side by side, stacked. Crazy. It's a lot. There are a lot more things that the kidney is doing. You guys were catching on to a lot of them. Here, let me start to share my screen here. You guys caught on to a lot of these already. Regulates water balance. As it's filtering water, okay, it's going to filter out 180 liters from your blood per day. 180 liters. You have 5 liters. So do you pee out 180 liters per day? I just like to make sure that you guys get those obvious points. Since we're not peeing out 180 liters per day, we're not constantly just, sipping on water 100% of our day and then peeing as we walk. It just doesn't work like that. What's going to happen to the large majority of that water that is filtered? If you're filtering 180 liters, but you don't pee 180 liters, what's going to have to happen to all of it? Perfect, perfect. Maybe you're not good. You guys all, I'm sure remember this, but remember filtration means it's going from the blood into the lumen of a tube. So we already saw filtration when we were talking about, it doesn't have to go into a lumen of a tube, it can go into the interstitial fluid, but when it leaves the blood, this is filtration. So when we're filtering 180 liters, that means that we have 180 liters per day leaving our bloodstream. So we better get most of that back into our blood. Okay? Just first bullet point right here. It helps regulate your water balance. Why? Because if we need to try to increase our blood volume, are we going to try to reabsorb more water or pee out more water? Reabsorb more or pee out more? See? Easy peasy, right? What if our blood volume is too high, or what if our blood pressure is too high? What are the kidneys going to try to do to water? Pee it out. Same thing with the next bullet point. Regulates ions. It's all about homeostasis. What if the body has too many sodium? What's the body going to try to do? Get rid of it. What's the primary way we can get rid of salts? Pee it out. Make sense? Or if our blood needs more sodium, or if it needs more potassium or whatever, the kidneys are going to help decide. Bicarbonates, right? That's an ion. Phosphate ions. Calcium. The kidneys are going to decide what comes and what goes. Regulates blood pH. Exact same idea. If your blood has too many hydrogens, we already saw that a major player is the respiratory system. If you didn't write this down before, even if you did, remember about 70% of all of your blood pH is regulated by your respiratory system. So what's doing the other 30%? Your kidneys. So if your blood has too many hydrogens, what are the kidneys going to try to do? Pee them out. Does that make sense? Is everybody on board? Homeostasis. It's just a gigantic homeostatic grid here. So if your kidneys go out, holy moly. Your kidneys start to fail. You stop regulating water properly. You stop regulating ions properly. Your blood pH goes out of whack. You guys see how that would very much impact the rest of the human body? All the rest of your organs are dependent upon proper function of your kidneys. Yeah, exactly, Tariq. Eliminating metabolic waste. Your body's constantly making metabolic waste. Every chemical reaction it does, there's a metabolic waste, like urea, so forth, so on. Exactly, Melissa. But that's a lot of that's to get rid of all the metabolic waste that's building up, and then reestablish normal levels of water ions and blood pH for sure. Regulates blood volume and pressure. That is directly linked with water balance, of course. And the last bullet point we can't ignore either, right? Endocrine functions. These don't just regulate water ions and pH, they also are super involved in red blood cell production. So somebody has renal failure and their kidneys are no longer producing erythropoietin, or renin, mother effing renin, right? Calcitriol, all about calcium. Blood calcium levels rely on calcitriol, vitamin D3. I hope I'm getting you guys to see the importance of these little things. There's not much more that does more than kidneys. All right, cool. Now, that being said, we're going to be diving down into the anatomy of the kidneys, and we're going to kind of do this between both lecture and lab, kind of how we've been doing. But I'm just going to give you the first layout of the kidney using this diagram right here. Actually, let's go one step back. I'm sorry, one step back. I want to get you guys to see how simple the urinary system actually is. There are only 4 organs in the urinary system. So in terms of organ simplicity, kidneys, ureters, urinary bladder, urethra. Oh, I forgot my markers over here. I'll be right back. So in terms of the simplest flowchart on the face of the planet, right? What's going to end up happening here? Oh yeah, definitely, always. It's not too hard. It's semi-mobile because it's just a DocuCam thing. It's an overhead. All right, but let's see here. Where's my pen at? It's right here. So filtration is going to be going on in the kidneys. Well, there's a lot more that's going to be going on in the kidneys as well. But kidneys is where it all this is where all the work is happening. Once all that work is done, 180 liters are going to be filtered. Well, we don't pee out 180 liters, so the large majority of that's going to be reabsorbed. All that's occurring here in the kidneys. Some metabolic waste is going to be secreted into that system. Nice. Now, at the end of the road in the kidneys, whatever's left over is going to be in your pee. End of story. As it leaves your kidneys, it's going to go into just a gravity-fed tube called the ureter. That gravity-fed tube goes from the kidneys all the way inferior to the bladder. The bladder is just a muscular sac. It's surrounded by a muscle called the detrusor muscle, and it's going to have baroreceptors that are monitoring for stretch. And as that bladder fills up and the stretch starts to push against the walls of that bladder, we're going to get a reflex. You guys are going to learn. That's going to cause the bladder to contract. Now, do you want to just pee yourself? I hope the answer is no. So, guess what? We're going to have some sphincters in between. And guess what? The sphincters in between are exactly the same as your digestive system. So between the bladder and the end of the road, we have a tube called the urethra, and that urethra is going to end in a urethral opening. Somewhere between the bladder and the urethra, and the end of the urethra opening, there are 2 sphincters. There's an internal urethral sphincter and an external urethral sphincter. Who remembers internal? Is that smooth or is that skeletal? Skeletal. You guys got it. So what's going to happen is when we get the reflex that causes the bladder to contract, that internal urethral sphincter is going to relax. And there's an external urethral sphincter that's going to contract. And we have to physically, mentally be aware of the relaxation of the external urethral sphincter to allow us to urinate. Make sense? It's exactly the same as the digestive system. Something I love about the renal system is that you get to apply everything you've been learning about. Yeah, exactly. Yeah. If you've given natural birth, then there's potentially some damage to the system down here. For sure. And you guys are going to see why when we talk about the anatomy of the urogenital diaphragm. You'll see where these muscles are located, and if a watermelon's got to go through there, and then what it does to the muscles of the area. Super simple flow chart. Now you guys are going to be learning a little much more complex flow charts of the kidneys, because this is where the function happens. This is where the physiology happens, right? So when we're talking about the function of the urinary system, we're going to be honing in on the kidneys. So the large majority of the rest of this unit, boom, we're just going to be talking about the kidneys, and more kidneys. Okay? So again, just so you guys can see that flow chart, kidneys, they're retroperitoneal, meaning that they are behind the peritoneum, of course. They got lots of blood supply you guys are going to be learning about going in through that hilum of the kidneys, just like there was a hilum in the lungs. And then this is where filtration, reabsorption, secretion all occurs. And by the time it's all done, we have pee. And that's going to empty out into the ureter, drain down, gravity fed only. That's why when you wake up and then you sit up and get up out of bed, all of a sudden it's like, man, I gotta pee. Because all of that urine that was up here and throughout the ureter, just lying there stagnant, it all goes right down into the bladder. All right, when you stand up, and then that extra stretch is going to get that urinary bladder to contract, at which time out through the urethra, and we'll be looking at the urethra on both the male and the female. All right, cool. Let's get down to the kidney itself, because I really want to get into some physiology today. I don't want to putz around too much. So here is a cross section of the kidney. It's just a coronal cut. So, side to side, if we just took this and were to cut from the patient's ear to the patient's ear, right? Coronal, crown. So we're cutting off the anterior section of the kidney. And when you do that, you'll notice that there are pretty distinct landmarks. The first major landmark are these crazy, almost triangular shapes here. Yes, no? Everybody see those okay? Let me, where is it? Over here. I am going to pull up a model as we go here, because I want to make sure that you guys see that these things are actually three-dimensional. It's hard when you're just looking at pictures sometimes to be able to see the three-dimensionality of the model. Urinary lab model, drag and drop, probably, but I know that they are in here, 100%. 100%. 60% of the time, I'm 100% right. It's a great saying. Always cover your butt. Exactly. All right, so you guys are going to be going through and looking at the anatomy of your kidneys, but this is a good one. This one will work really well. So do you guys see that triangle that I was pointing out? Kind of looks like this when it's two-dimensional, but three-dimensionally, it's actually almost like a hot air balloon. So you got this pyramidal shape. It looks like a pyramid. That's why these are called the pyramids of your kidneys. They're actually called the pyramids. They're just a big landmark right in the middle of all of this. Okay, so there's a three-dimensionality that's going to go along with it. These are all called the renal pyramid. I'm just pointing those out because they're a landmark. They're really easy to spot, and they're going to get you guys started on your anatomy of the kidneys. Now, that being said, there are two major regions of the kidneys. There is a cortex, which is this entire surrounding band. Okay? The cortex is surrounded by a capsule. So this outer layer that you see right here, let me go up maybe to this portion. This outer layer right there, that is a hard shell. It's called the renal capsule. It's a very hard shell. The second you penetrate the renal capsule, you're now in the renal cortex. And the renal cortex is going to go all the way until you hit the pyramids. Okay, so the renal cortex is this section right here. Everybody clear on the renal cortex? Please write down renal cortex. What I want you to do is I want you to say 100% of filtration occurs in the renal cortex. 100% of filtration occurs in the renal cortex. So everybody, where does filtration occur? Renal cortex. Pretty important spot. And what you guys are going to do is you're going to be learning what exactly allows for the filtration up in there. All right, so the next region right here is cortex. Everything from here, from the end of the cortex, all the way down to this tubing you see in here. So all of this region right here, this is called the renal medulla, right? And medulla, you've seen through the entire semester, is the core of the organ. So all of this. So basically the pyramid, but it's not just the pyramid. It's actually this space in between the pyramids as well. These are called the renal columns, and the renal pyramids are the triangular structures. And those two things combined all the way around is the renal medulla. Okay, everybody clear on that? We'll see some of the function of the renal medulla, but a lot of it has to do with blood supply through the renal columns. And then we're going to see that the renal pyramids have a double function as well. Okay, cool. Now, filtration's occurring in the cortex. A bunch of magic's going to happen. You're going to have to learn all of that. Filtrate is going to, hopefully, most of it's going to be reabsorbed, but some of it is not going to get reabsorbed. Whatever doesn't get reabsorbed is going to make its way through the renal pyramid, and it's going to end up down in these lighter ends. You guys see how it goes from a darker area to a lighter area right here. So just like what you guys have been seeing through a lot of the semester is that if you have a, a nipple-like shape, then it's called a papilla, right, or papilla. We've seen all sorts of papilla throughout the semesters, including papillary muscles in the heart. So this down here is called the renal papilla, renal papilla, renal papilla. And the renal papilla is pretty cool on your models. I love it. Okay, so if we look in, oops, come here. If we look at how this all works, you'll start to notice there's actually some tubes draining down through that renal papilla. And when we look at the end of the renal papilla, three-dimensionally, that's what this structure right here is that I'm circling. This is just the very, very cap of the renal papilla, just, turned so we can actually see the end of it. Do you guys see how there's all these little holes in it? Yes, no? So those are going to be the tubes that have been draining through this renal pyramid, and they're going to end up draining and anything that's not reabsorbed into this tube system here. Okay? So that's the renal papilla right there. I love it. I love that. And you can see it from this view as well, this lighter region right here. So where does all of this drain into? It drains into this tube system. Now, it doesn't go straight to the ureter. We're going to go through some smaller tubes to get to some bigger tubes to get to a larger storage compartment into the ureter. So where does it go? It goes from the renal papilla, right here, and it's going to go into this portion of the chamber first. Okay, these are smaller, so they call them minor, and they're called calyx. Calyces is plural, calyx is singular. So this is a minor calyx. Multiple minor calyces will drain into a major calyx. And then you got a major calyx over here and a major calyx over here. And all of that is going to drain into the renal pelvis. Okay, so this big chamber right here is the renal pelvis. Those three things together, the minor calyx, the major calyx, and the renal pelvis, that's all called the renal sinus. So basically, once your filtrate enters the renal sinus, we can't do anything else with it. At that point, all we can do is pee it out. So everything that enters the renal sinus becomes part of your urine. Make sense? All right, so let's do this again. I'm just being redundant here. Filtration occurs in the cortex. Some magic is going to happen. We're going to have filtrate go down and up, and it's going to do all sorts of crazy stuff here. But in the end, it's going to end up draining down through the renal pyramid. It's going to make it to the renal papilla. It's going to drain into the minor calyx, which drains into a major calyx, which drains into the renal pelvis. From there, it's all going to drain into the ureter. Back to a flow chart, right? So if we want to just take and look at your kidneys, we can actually expand upon this idea of your kidneys, where we have filtration that is all occurring in the renal cortex. Some magic's going to happen. You're going to have to learn. It's much more complex than just what we're showing here, but that's going to end up draining down through the renal pyramids. That's going to drain into the renal papilla. That's going to now drain into the minor calyx, which drains into the major calyx, which now drains into the renal pelvis, which now drains into the ureter. And then you guys, urinary bladder. I want to make sure you guys know this is urinary bladder because the body actually has multiple bladders. And then from the urinary bladder, urethra. And you guys are going to be learning urethra, a few more portions of the urethra, a few more portions of the bladder, so you'll be able to expand upon those parts of the flow chart. Everybody feel pretty comfortable with learning these words and kind of having a basic idea where they are in the kidneys already? Awesome. Super duper cool. So now if you go in and just look at a blank model, you might be able to start to see some of these things. Renal cortex out here, 100% of filtration occurs out here. Renal pyramids, renal papilla, minor calyx, major calyx, renal pelvis, ureter. Yeah, it's pretty straightforward for now. What we're going to now start to do, and we got to get there right now-ish, is we're going to already start to look at the renal cortex, because this is where the large majority of the physiology occurs, in conjunction with a little bit of the renal pyramid. So everybody, look at the model, and you'll notice there's something that looks, I don't know, like little seeds, like little pods. Lots of blood supply going to these things. Now you can also see some white tubes. These white tubes diving down, going around, up and around, ziggy, 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 what the, okay? This right here is where the physiology happens. So this is where the most of your attention needs to be placed. These little round circles here is where 100% of filtration occurs. It's these little round circles. These are called glomeruli. And this whole system, you guys don't need to write that down just yet. I'll come back to that. The whole system where we have one glomerulus, glomeruli is plural, you got blood supply into that glomerulus and you got tubes leading away, coming back down, coming back up, going into a big drainage tube. This right here is the function, the primary role, the physiology of your kidney. It's called a nephron. This is why a kidney doctor is called a nephrologist, because it's the study of nephrons. Everybody, if I were to ask you what anatomical unit is the functional unit of your kidney, what's the answer? Perfect. The answer is the nephron. You guys must know all about the nephron. This is like 90+% of your exam will be on the nephron. And your comprehensive final will have tons of stuff on the nephron as well. Greater than 1 million per kidney. Now, that's a lot of nephrons. Where do we find the first portion of this nephron, this little ball that you guys see right here? What part of your kidneys, what region of your kidneys do we find this little ball? Always. Good. That's always in the renal cortex. Always. If you see histology models, hint, hint, and you see this little round circle, hint, hint, where do you know you are at? You know you're in the renal cortex. End of story. Stop overcomplicating things. All right, cool. Now, this is where 100% of filtration occurs, and we filter 180 liters per day. So a major function of the nephron is filtration. And structure is going to determine function. So we're going to talk about the anatomy of that nephron and how it promotes filtration. Here, out here in the glomerulus, but not anywhere else. Again, though, we filter 180 liters per day, but we only pee about 1.5 liters per day. So what's happening to the large majority of it? It's gotta get reabsorbed. Okay, 178.5 plus or minus liters of material are going to get reabsorbed, and that's occurring all throughout this system. Now, the anatomy changes as we go through this system. So what do you know about the function of the reabsorption? If the structures change, then the functions change. So different portions of this crazy nephron stuff going on here is going to reabsorb different material. Yes, no, maybe so? It's all going to be based on what? Ooh, I love this question. If we're talking about absorption, reabsorption, if we're talking about secretion, the exact same question I asked in the digestive system 1,000 times, maybe not 1,000, but you guys get the point. If we're talking about reabsorption, absorption, and if we're talking about secretion, what do we know we're talking about? What's the physiological importance of saying these things? Andy, you got it. Does everybody remember me saying transporters, 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 transporters? We're talking about reabsorption, we're talking about secretion, we're talking about absorption, it doesn't really matter. We're talking about moving materials across the cell membrane, then we must be talking about protein transporters. Yes, no, maybe so? So if I'm going to reabsorb water, I better have some functionality, some structures that allow for the reabsorption of water. If I'm going to be reabsorbing sodium, boom, I better have some transporters for sodium. If I'm reabsorbing glucose, if I'm reabsorbing potassium, calcium, bicarbs, whatever the case may be, I better have transporters for those. And the transporters through the system change because structure changes and structure determines function. Same thing goes with secretion. Secretion. There are some materials that are too big and too charged to be filtered. End of story, but we still need to get rid of them. This is how your body gets rid of all penicillin. If you've ever taken penicillin, it makes your pee stink real bad. Is that we're not going to filter it here. We're going to have to secrete it from the blood back into the tubes out here. But again, if I'm talking about secretion, I'm taking something from the blood and I'm going to move it into those tubes. I need protein transporters for that function. Make sense? And finally, at the end of the road, whatever doesn't get reabsorbed, everything that's left over will be excreted. We're going to learn a math problem for that. We can actually do that, but I don't know. We can do that right now. Yep, 1.5 liters approximately. Just depends on the patient, but yep, you got it. So what I want you guys to do is this is such an easy, I don't know about easy, but this is a lesson plan that, one, is very important to the system, very important to this unit. But if you start to think about how this system works, we want to talk about the function of the nephron. Let me come down here. I'm not zoomed out quite far enough. If I really want to sit down to study the nephron, we have an equation that can walk through this list, right? So what were those 4 functions of the nephron? We could write it down as a list. Filtration. That's the first thing that's going to happen. First thing that's going to happen is filtration, right? 180 liters per day. First thing that's going to happen at the very beginning of the story is we filter. Now, once we start filtering, we're going to immediately start to reabsorb stuff. I'll give you guys an analogy of what the kidneys do. It's crazy. Absorption. I'll give you guys an analogy. The way the kidneys work is like the TV show Hoarders. Anybody ever watch that TV show? Your body is a hoarder, all right? It holds on to all sorts of shit. And in the kidneys, what's going to end up happening is it's just like that TV show where you go in and they, you know how they just pull like everything out of the house and they just pile it on the front yard and in trucks and stuff like that? And then they start to pick through it. And they say, oh, I'm going to keep this. Oh, I'm going to keep this. I'm going to keep this, right? And if you're like a true hoarder, they end up wanting to hold on to everything that they've thrown away. You guys know what I'm talking about? I'm only getting Tariq response right now. That's the kidney. It throws everything away, but then says, no, I can't get rid of that. No, I can't get rid of that. You guys are going to see how this works. It literally filters everything out, 180 liters per day, and then it's like, nope, I'm keeping you, I'm keeping you, I'm keeping you, I'm keeping you. That's reabsorption. Another way to look at it is that you have the dirtiest desk on the face of the planet, and you take your arm and you sweep 20% of all the material from that desk into a garbage can. Just take and sweep the dirty, I mean, piled high, feet high of junk and stuff. You take your arm and you just sweep 20% of that into a garbage can. And then you go back to that garbage can and you pick 99.9% of the crap you just swept into the garbage can, you pick it back out and you put it by hand back onto that desk. So that in that garbage can, there's only 0.1% of the original sweep. That's your kidneys. It's crazy. Reabsorption. Okay, we're reabsorbing the large majority of this, 178.5 liters per day better be getting reabsorbed. Now, there's a very negligible amount in terms of total volume, but there is some material that can't be filtered. It's just too big, it's too charged, can't make it through the filtration system. So this stuff has to get secreted. And we know the word secretion is all about taking it from your blood and putting it inside of a tube. It's going to need protein transporters for that function too. Yeah, exactly, Tariq. Or the parents that say they're going to throw away the toys, but they just secretly put it in a box and then like give it to the kid for Christmas again next year. What? No parent would ever do that. 4, the final part of this nephron is going to be related to the top 3 functions. If we filter 180 liters per day, but we reabsorb 178.5, we secrete just a tiny fraction, negligible. Like, you know, we're not even putting it into our calculation, negligible. Whatever's left over at the end of the road, that gets excreted. Okay, this is going to go through a process of urination. So if we're thinking about filtration, which one of these promotes filtration? Does filtration promote filtration? Go ahead, guys. Does filtration promote filtration? Sure, it's taking it from the blood and it's pushing it out into the tube system. So that promotes filtration. Does reabsorption promote filtration or oppose filtration? Remember this from Unit 2. So this is in opposition. Does secretion promote or oppose filtration? If it's taking and moving it from the blood into the tubes, then it's promoting filtration. Good. And whatever gets filtered at the end of the road, that's what's going to get excreted. So let's take this and say excretion, whatever we have at the end of the road, well, it's a property of what gets put into the system to begin with, which is 180 liters per day. We got to filter first. But the large majority of what gets filtered is going to go back into that bloodstream. So we take filtration, we get rid of the reabsorption, and all we're going to do is we take and add back the smallest little value of secretion. Now, that will go into our excretion, but you guys will never have to calculate it for it. So this is the equation that will help you guys understand the function of the nephron. So from here to the end of this unit, anytime you guys go to sit down and study the nephron, write this equation down, okay? Thank you. So what are you guys going to have to do? The same thing you've been doing all semester. We write down our equation, we write down our list, we put down our diagram, and then we ask ourselves, what one word should you be asking yourself as you study? What one word? Love it. I'm hoping I'm making an impact on you guys. The answer is how. The word that you guys should be saying is how. How does filtration work? How does reabsorption work? How does secretion work? And it's always structure determines function. That's why we're teaching anatomy and physiology. Yes, no, maybe so? Cool. All right, so what we're going to do is we're going to look, goodness gracious. At the structure of the nephron, and we're going to learn a way to draw it that's going to help us simplify the structure. It's we're going to be drawing it in this linear fashion. Now, I want you guys just to look at the screen. Don't try to draw anything or write anything yet. Don't even read anything, please. Just follow along. This up here is that glomerulus. So this is where 100% of filtration occurs. Filtration goes from the blood into your tube systems. And now it's going to flow, this filtrate's going to flow through the tube system. And as it does, it's going to be getting reabsorbed back into the blood. The way it really looks is more like this. See how these tubes are twisty, windy, crazy, convoluted? See it just like almost impossible to follow the tube with your eye. You have to kind of like really take and focus and get through that maze. You're not going to want to draw this. You're not going to want to draw this crazy tubular mess, are you? I hope not. So what we do instead is we draw it out as this really nice linear format. Not like this. Like this. Is everybody clear just visually what we're working on? When we go to draw the linear format though, I need you guys to kind of make sure you remind yourself constantly that it looks more like this. It looks more like a gigantic mess of spaghetti noodles that are all twisted together. But we are going to be learning it in a linear format. So let's do that. And then we're going to be talking about the function of each one of these. Yep, yarn, string of yarn, you got it. So the way we're going to do this, of course, is just straight up drawing it. That's always the best route to learning this material. Okay, yeah, I'll go ahead and draw here. That's fine. Where we start with is we want to make sure we understand that filtration requires blood and that this is 180 liters a day. We got to remember that this is 180 liters that came from your blood. Okay, so we're going to have blood supply into this system. Does that make sense? We're going to be learning all about this blood supply today. We got a lot of blood supply to learn. A lot of blood supply to learn. So when we go to draw it out, I recommend drawing it out kind of with this flow of information where blood is going to be entering this system through a tube, right? So we got blood coming into this system. Now, by the time it makes it out here to the nephron, it's the smallest tube possible that still has smooth muscles. So which blood vessel, yeah, Tariq, you got it. Which blood vessel is the smallest as we've been diverging and diverging and diverging? Which is the smallest but still has smooth muscle? Just unit 2 stuff, y'all. Arteries turn into what? With smooth muscle that surrounds circular bands of smooth muscle so we can control it. Arterial, right? This is an arterial, and it's going out to the nephron. So it's going to be called an afferent or afferent. I like afferent because A, and I like efferent because E, but it's actually pronounced afferent and efferent. Those words just sound so similar that I end up saying afferent. And this is an afferent arterial. The arterial port is very, very important because arterial tells us smooth muscles. And smooth muscles tells us that a nervous system must be communicating with it. And if it's an arterial, now we're talking about adrenergic receptors. Were you guys able to follow me on that? Awesome. Now, this blood is going to flow into this little ball that I pointed out quite a few times. We'll talk about the different adrenergic receptors, Tariq. There's going to be alpha 1 and beta 2. So this is going to go into that little ball that I've been talking about a few times, okay? So we're just going to draw the ball. We want to make sure that we leave a gap, a space for a tube to leave, both on the top left and here in the middle off to the right. Okay, the tube that leads off from the top left is more blood that's leaving. So blood comes in and blood goes out. So if blood's going out, it's going out through a small tube surrounded by smooth muscle. So again, we have another arterial up here. But this arterial is leaving this nephron. So it's going to be efferent. Make sense? And in between, we want to connect these things. Now, I'll show you again on the picture what's actually happening. This looks, literally, I just say spaghetti. Right? Check out this really cool structure in here. Kind of almost looks, again, like a ball of yarn. I like that analogy for the glomerulus is a ball of yarn because that's literally what glomerulus means in Latin. It means ball of yarn. And you can kind of think of it like a fist as well, because it's kind of like these big knuckles, these big fingers that are wrapped back around. But we don't want to draw this. I hope you guys are on board with me on that. I can't draw that. So instead, what I will do, where am I here? I'm right here. What I do is I just end up drawing a little horseshoe, basically. So just wrap it around, leave yourself a little bit of room. You don't need a lot of room. It doesn't need to be perfect. Blood's going to come in. Blood's going to go into this little, ball of yarn, and then it's going to exit. So that's all you need. You just need to make sure that you understand that as blood comes in, it is going to go out through a big ball of yarn, but we can't draw that. So we just draw something simple. And this structure in here is where 100% of filtration occurs. That in there is called the glomerulus. Glow, mer, u, lis. That's the glomerulus. This little circle that I've referred to a number of times is actually a hard shell. It's a capsule that surrounds that glomerulus. Okay, this capsule, some people just write down nephron capsule, which is fine, but most doctors aren't going to refer to it as a nephron capsule. They're going to refer to it to its original name, which is Bowman's. Okay, Bowman's capsule. That's the name of the anatomist that discovered it. So I got a Bowman's capsule and I got a glomerulus. So the glomerulus is a modified capillary bed. It's modified, then different than the other capillaries that you've been looking at so far this semester. The other capillaries you guys looked at just had cracks between simple squamous epithelial cells. If you recall, there were just cracks. Whereas the glomerulus is modified because it's more like that other analogy that I used, where if you take your hose and you just start stabbing it with an ice pick, you create all these tiny little holes called fenestrations, right? You guys remember those terms maybe? Fenestrations? It's okay if you don't. Filtration is occurring in normal continuous capillaries between cracks. Yep, that's what fenestration means. You got it. Between cracks, because basically there's just not, ultimately there's not a lot of filtration that's occurring out in your tissues relative to your kidneys. Whereas here, we need to have a massive amount of filtration. So we just put holes in there, literal holes. Because what can go through a hole? Anything small enough. Make sense? So here in the glomerulus, this glomerulus is fenestrated. We're going to see a slide for this. We're going to actually talk more about the anatomy of the glomerulus. But when filtration occurs, that filtrate now is going to be moving from the glomerulus into the Bowman's capsule. So it goes into this capsule here. And it can stay in this capsule because the capsule actually has a serous membrane. Okay, it's going to have a membrane that, one, is adhered to the walls of the Bowman's capsule out here. So that sounds like a parietal serous membrane, right? So this is parietal out here. And then what do we know? There's always going to be one that's on the organ itself, and that one's going to be visceral. Cool? This will help you guys with your lab lab objectives quite a bit. So we've got a parietal layer and a visceral layer. They're just called parietal and visceral layers. There's not like a fancy name like there is pericardium or peritoneum or pleura. So I hope you guys are seeing filtration, filtration, filtration. This is a pretty fancy little unit here where glomerulus is going to be filtering all 180 liters into the Bowman's capsule. And so we have a name for these two things together, which is called the corpuscle. Okay, this is going to be the nephron corpuscle or glomerular corpuscle. Like muscle, it's got this weird S-C-L-E, glomerular corpuscle. Over 1 million, Tariq, over 1 million per kidney. Sorry, I got a crying baby in the background. She wants to boogie board, apparently. I'm not joking. That's what's going on. This is all cry over a boogie board. Okay. The waves are nice down here. Okay, so let's go back to the lecture because I just want to tie these things all together for you. Right? So blood in through the afferent arteriole. It's going to pass through the glomerulus, and as it does, the glomerulus is going to be able to filtrate. We're going to talk all about this filtration that's going to occur and what's going on, what's being added to the filtrate, blah, blah, blah. But that filtrate is going to go out into this capsular space created between the visceral layer and the parietal layer of the glomerular capsule, the Bowman's capsule. Exact same thing as glomerular capsule, okay? So if you see glomerular capsule, I'm talking about the Bowman's capsule. Whatever blood does not get filtered is going to leave through the efferent arteriole, or efferent. Does everybody feel comfortable with what we said so far? There's a lot more to learn, so I just want to make sure you guys are comfortable with each step. How can we filter so much here? One, fenestrations. I've already kind of talked about those. But two has to do with another structure determines function. Anybody notice anything? Yeah, exactly, Tariq. Does anybody know anything or notice anything weird about the afferent and efferent arterioles? You're okay. No, not a color. They're both the same. They're both red because this is not your typical capillary bed. This is not where gas exchange occurs. Yeah, the size and the thickness. Actually, Ruju, I love that you said that because the thickness does matter. The afferent arteriole has some extra cells around it that you guys are going to be learning about. But the primary difference you'll notice is the diameter. Afferent is bigger, efferent is smaller. You go back to my diagram, afferent is bigger, efferent is smaller. You should always draw it this way. You should always remind yourself that afferent is bigger, efferent is smaller. Why is this important? What do we know about the diameter and blood flow? If the diameter is bigger, resistance is smaller, and blood flow is increase, decrease, no change. Blood flow. Will it be increase, decrease, no change through the afferent? We're not talking about blood pressure, Andy. We're talking about blood flow. So, yep. Nope, yep. No, not increase through efferent. So the larger the diameter of the tube, remember blood flow is a pressure gradient divided by resistance. Yes, no, maybe so? You guys remember that? So we're talking about blood flow. This is a pressure gradient that's typically created because of cardiac output. In this particular case, it's going to be created due to mean arterial blood pressure. The primary thing that controls mean arterial blood pressure is still cardiac output. And in terms of blood pressure inside of these tubes, but it could be blood volume. And then we'll be talking about the resistance here. Over resistance, right? So if I have a larger diameter, what's happening in my resistance? Increase, decrease, no change. So as that resistance in the afferent arteriole drops, what happens to blood flow since they're inversely proportional? Did that help? Did this visualization help anybody? I hope so, right? I'm trying to use what you've learned before to make you understand this a little bit better. So if I have more blood flowing in, well, let's talk about the efferent. What's happening to blood flow out? Increase, decrease, no change, because the efferent's smaller. So more blood flowing in than can flow out. So what's going to happen is it's going to build up a dam in here, right? And the pressure inside of the system is going to go up a lot. Does that make sense to everybody? More blood in than can leave. Therefore, the pressure is going to build up. And as that pressure builds up, we're going to create more of a pressure gradient to be pushing our filtration, hydrostatic pressure inside that tube. Don't worry, you don't need to write any of this down now. We're going to do it in a little bit. And that's going to allow for a greater amount of filtration, in addition to fenestrations. So now we got this higher pressure gradient, and we have fenestrations. Man, we are flowing 180 liters per day because we got a bit like we got a French press going on here. We're literally pushing our blood through this system. Make sense? Ish? We'll do it so many times, guys, that it will start to make sense if it's not making sense just yet. Cool. Awesome. We're going to draw some other stuff in just a bit with the efferent arteriole, because this actually leads to a capillary bed that we'll talk about. But let's go ahead and finish off our tube system. In order to do that, I'm just going to switch to a new page, and I'm going to teach you guys how to draw things pretty quick, because you don't want to waste your time as you're going to study these, but you're going to be wanting to draw this nephron over and over and over. You really, really want to get into a habit of being able to. Where's my focus button? It changed it all on me here. Here's a good focus button. Haha. You want to get into a good habit of being able to draw this nephron. So make sure that you practice repetition on it. So you're going to just have blood flowing in to a circle. You want that leaving off one side, which we're going to get to next. We want a tube that's bigger going than coming out. Boom. Right? We're going to have ourselves our little horseshoe shape. Don't have to change colors to make this work. Now we have ourselves an afferent arteriole leading to the glomerulus, glomerulus leading to the efferent arteriole. We have a Bowman's capsule. The combination of the Bowman's capsule and the glomerulus together was the glomerular corpuscle. Boom. So keep working on that. Try to get it drawn out, and then, you know, the labeling can come after. So now, as filtration occurs, all this filtrate's going into this Bowman's capsule in the capsular space. It is a hard-shelled capsule. It's not going to be able to go back into your bloodstream, and we just have one road that can allow this to leave. Right? Everything's trying to go from an area of high pressure to low pressure. So now the pressure inside this Bowman's capsule is building up. Well, we give it a way out. Make sense? These tubes right here are crazy, right? As we go afferent arteriole in, glomerulus, filtration out into the Bowman's capsule. There's only one place for it to go. It's into this tube right here. This tube is crazy, twisty, windy. It is massively, massively convoluted. Okay, it takes so many twists and turns. It's a very convoluted tube. It's also a very small tube, so we don't call it just a regular tube. We call it a tubule. And since it's close to or nearest the glomerulus, it's attached to the glomerulus, we call it proximal. Do you guys remember those that terminology for your arms and your legs? If it's closer or attached to your body, closer to the attachment to your body, we called it proximal. And what's it called when it's far away? Distal. So if you follow this along, you're going to see the proximal convoluted tubule is going to end up descending. We'll talk about this called the nephron loop, or the loop of Henle. But now we come back up on the other side from the loop of Henle, and we have another convoluted tubule. This one is not attached to the glomerulus. So we're going to call this portion out here the distal convoluted tubule. Does that make sense? So let's draw it out. The way we can draw this is, I just like to simplify it. I don't want just straight line, but I want to make sure that I understand that this is convoluted. So I'm going to go up, I'm going to go down, I'm going to go up, and I'm going to go down. So now I just follow that. Now I got myself a convoluted tube, right? It's twisty, it's windy, it's attached to the glomerulus and the Bowman's capsule. Sweet, I got this. This is the proximal convoluted tubule, so I can abbreviate it as PCT. On a lab practical, don't you dare abbreviate. But on a lecture exam, you're always welcome to abbreviate for me, okay? Unless I specifically ask in the question not to abbreviate. But yeah, lab practical is, you're getting tested on the name, so don't abbreviate, please. So what's going to end up happening is all of this tube system you've seen so far, this is all up in the renal cortex. So I'm going to put in a dashed line so you guys can see this a little bit better. But everything above this dashed line, everything above it is going to be renal cortex. So everything below it's going to be what? The medulla, and more specifically, it's going to be passing through the renal pyramid. Okay? You're not wrong. It is definitely renal medulla. But where the functionality happens, where the nephron goes, it goes into that renal pyramid, not into the renal column. So that's why we want to specify. So now what's going to end up happening to this tube is it's going to drop from the renal cortex. It's going to end up descending down into the renal pyramid. And as it does, it's going to change its shape. What do we know? Structure determines function. So this is going to descend down before looping back up again. And as it loops back up again, it's going to change shape again. I'm kind of getting a little bit big here, but this will work. Everything you guys see down here in the renal pyramid is called the loop of Henle, or the nephron loop. Loop of Henle. Name of the guy that first found it. You can call it nephron loop on your lab practicals and whatnot. That's okay. I'm not going to mark you off. Loop of Henle is what I know it as. Therefore, that's what I'm going to refer to it as most of the time. Now, the loop of Henle has quite a few different, portions to it. You can see that. The structure is changing from here to here, and it's changing as it goes back up this way. So the first way we want to think about the loop of Henle is one portion of that loop of Henle is descending. Okay, it's going down from the renal cortex, and it's diving down into this renal pyramid. So this is going to be called the descending limb. So if on a lab practical I were to say, what specific structure is this? You're going to say, right now, what you've learned is this is the descending limb of the loop of Henle. You got to say it all. You can't just say descending limb, and you can't just say loop of Henle. This is the descending limb of the loop of Henle. Does that make sense? And over here is going to be the ascending limb of the loop of Henle. So as it goes back up, we got ourselves the ascending limb. The next thing you'll notice is that there's certain portions of this loop of Henle that are thick and there are certain portions that are thin. So that's the last part that you guys will need to add to the name. So if I put a pin right here, said what specific structure is this? You're going to say thin descending limb of the loop of Henle. Whereas if I put it up here, you'd say thick descending limb of the loop of Henle. Or if I were to put it here, you'd say thick ascending limb of the loop of Henle. Everybody understand the combination of terms? Awesome. Thanks, y'all. All right. Now, we'll talk more about the function of each one of these in a bit. Let's just get it drawn out. As it comes back up, right? I'm going to go ahead and change the color here because I don't know why I did that, but whatever. So now as it comes back up, this is part of the distal, but it's convoluted, so we better put some convolutions in here. So we got ourselves a distal convoluted tubes. And those distal convoluted tubes are all going to end up going into a huge drainage system. And I'm going to have to put my drainage system in a little bit crooked because my terms. And that's all right. Okay, this drainage system is going to be collecting from many other distal convoluted tubules. Okay, so this over here is the DCT. There's actually a portion that connects from the DCT to this big tube right here called the collecting tube, the collecting tubule. And that's all going into this big tube. And this big tube, you see, goes down into that renal pyramid, and it's going to go all the way down until it hits the renal papilla. This one right here is called the collecting duct. Okay, and it's going to be having all sorts of collecting tubules that enter, lots of different collecting tubules that are going to be bringing whatever's left over from the system, it's bringing it into the collecting duct down to the renal papilla. And then from there, you guys should have a pretty good understanding of what's going to happen, right? Renal papilla, minor calyx, major calyx, renal pelvis, ureter, urinary bladder, urethra. Make sense? All right, let's go to some pictures real quick so you can see all of this. Just going to kind of show this in multiple different angles. Here, good start. Afferent arteriole into the glomerulus. Glomerulus filters out into the Bowman's capsule. Whatever does not get filtered goes into the efferent arteriole. The filtrate, the first portion of the filtrate from the capsular space of the Bowman's capsule is going to drain into the proximal convoluted tubule. The proximal convoluted tubule out in the renal cortex. There's some really cool pictures for that, right? Like this. So we got this tube system that's in the renal cortex, the light pink area, and it's eventually going to dive down into the dark pink area, which is the renal pyramid. This down here is going to be your loop of Henle, the nephron loop. There's a descending limb and an ascending limb, and there's a thick and thin portion of it. As it ascends back up into the renal cortex, twisty, windy, windy, right? That's your DCT, or your distal convoluted tubule. You got your collecting tubule over here that's all going to go into this big collecting duct. Yes, no, maybe so? We could do this so many different ways. Let me go ahead and go into this one. It's a little bit harder to follow it, but that's okay. You still got to do it. Afferent arteriole in to the glomerulus, efferent arteriole out. Whatever's getting filtered is going to go into the proximal convoluted tubule, up and over and twisty, windy, dives down. What I want you guys to notice about this particular picture is that the descending limb is on the right side. So what if I were to do here on a lab practical? What if I were to point to this portion right here? Then you need to be able to write what? What is this portion of your tubule system? Don't be reading the lecture slide. What is this right here? What specific structure is this? Right under my mouse. What's specific? Parker, great job. Did everybody else maybe just type it a little bit slower? Did you guys see this is the proximal convoluted tubule, dives down. So this is going to be the thin descending limb of the loop of Henle. Yes, no, maybe so? Okay, awesome. Thanks, y'all. Okay. You can do this on all of these different pictures. You can follow back up, DCT, collecting tubules, collecting duct. Please do this as many times as you need to understand what's happening. Go back to the model. So what you'll see now on this model is this exact structure. Okay, this model is really just blowing up here, but that right there is the glomerular corpuscle, blood in kind of thing, right? That right there is that Bowman's capsule and the corpuscle, afferent arteriole in. So this portion of the tube is going to be the PCT, descending, ascending limb of the loop of Henle, DCT, collecting tubule, collecting duct. Collecting duct drains all the way down, all the way down, all the way down to the renal papilla, into the minor calyx, major calyx, renal pelvis, ureter. Lots of other models showing the exact same thing that are zoomed in a little bit better. A lot easier to see. Please be able to follow. So for instance, this one right here, this one's going off to the right. So the descending limb is on the right side. The ascending limb's on the left side. Students get this wrong on lab practical all the time because they're just trying to read from left to right. It doesn't work like that. You got to follow the tubes. Okie dokies? Cool. And we also have models just of this crazy structure right here. This right here. Remember, how can you tell which arteriole we're dealing with? Size and, as Ruju pointed out, the thickness. You'll actually notice that this is not simple squamous epithelial cell. This is a simple cuboidal, which is very unusual for an arteriole. We'll come back to that. But yeah, bigger is afferent, smaller is efferent, is efferent. Come in, and boom, we got the visceral layer. Do you guys see how the visceral layer kind of looks all spiked and stuff? Kind of looks like, I don't know, this light color. This is the visceral layer. And these specific cells have a function. These are cells that cover the glomeruli, that cover the capillary beds. These are cells called podocytes, and we're going to be seeing them after our break. But filtration is going to occur out into the Bowman's capsule, and the Bowman's capsule is coated as well with a parietal layer. Boom. All of this together is called the glomerular corpuscle. Sugar coating of a donut. I'd eat that. I'd eat that. Here's another great view of it. Afferent, efferent, glomerulus. By the way, if I were to show this model, doesn't really matter, on a lab practical, and I stuck a pin in the red versus the white, you got to remember these models are peeled away. They're dissected away so you can see all sorts of different layers. No, this isn't the pyramid. The pyramid's here. Okay, this is a zoom in. Let me show you guys what's going on. This is the whole kidney. This is just one section, one piece of that pie. So it's one area that's got both cortex and renal pyramid. So that renal pyramid's right there. So what this final one is this is where 100% of filtration occurs. So we have to devote quite a bit of time to this little structure right here. So this is the renal corpuscle. You got it, right? This is one of these balls right here. Everybody with me on that? So if you're looking at this, now we got afferent arteriole in, efferent arteriole out. Now, this is peeled away, it's dissected. So if I were to put a pin in the red, that's the glomerulus. But if I were to put a pin in the white, these are cells, specialized cells. They cover the entire glomerulus, but they've been cut away over on this side. Is that somewhat clear? These cells are called podocytes. They look like they actually look like this. This is the real image of them. This is the surface electron microscopy. So they've coated this with this gold, and they've taken a picture of this underneath a microscope, a laser microscope that's worth about $1.5 million. These pictures are very hard to take, and it ends up showing you basically this diagram in more detail. So these cells are called podocytes. They're leg cells, leg. And they have these leg structures that wrap around this fenestrated capillary of the glomerulus. And those legs have feet, tons and tons and tons of feet that intertwine like laced fingers. And in between each one of these feet is a slit. Okay, these are called filtration slits. Yes, you do need to know about filtration slits. So filtration slits are found in between each of the foot processes of the podocytes. You can see them here. Those are the foot processes, and the filtration slits are all in between. All right, so if I were to, on a lab practical, ask you about these cells or the cracks in between these cells, the cells themselves are podocytes, and the cracks are called filtration slits. And it's these podocytes that make up the visceral layer of the Bowman's capsule. Whew. All right, let's take a break. Let's come back at 45 after. Okay, my friends, let's get rolling on this. Awesome. Thanks, Tariq. I'm hoping everybody else is here. Now you can hear my voice. Go ahead and do a quick poll. Aquí. Bien. I'll just do do this poll thing. So here, I'm here. I clicked yes. All right. One is joining. But yeah, Ariana, Brianna, Heather, Ruju, Sydney, you guys all around. Sabrina, you just showed up. Awesome. Just click yes if you're here. If not, I gotta wait a couple more minutes. Ariana, Brianna, Ruju, Sabrina, cool. There'll be a poll somewhere on your screen, but that works for me. I like the yeses. Sydney, Ariana, Brianna, Sydney. All right, well, I'm gonna go ahead and start. Let's actually go to the lecture. We'll see how long I can stay away from Chalk Talk. I love Chalk Talk so much, but I want to just always prove to you guys that what we are learning via Chalk Talk is exactly what the lecture, the PowerPoint, it's exact same stuff. Okay. The functional unit of your kidneys is the nephron. The nephron allows for filtration from a particular subset of its anatomy, reabsorption, secretion, and at the end of the road, whatever's left over, it will excrete. We've seen the anatomy of this system. I'm not going to go over it again multiple times. What I do want to make sure that you guys see is that the structure clearly changes as we go through this tubule system. Let's look at the epithelial tissue. What do you guys see different about the PCT and the DCT, for instance? What's going on here? Don't just say colors. The PCT has larger cells. Good. What do you mean by AFF? Cuboidal. Yeah, for sure. They're both cuboidal, but the PCT has larger cuboidal cells. Both are ciliated. Good. Let's hone in on that, Melissa. Do you guys notice anything about the cilia here versus the cilia here? Ooh, versus the cilia here. I said, don't you dare say color, Tariq. Dweeb. You got it. Where do we have the most cilia? Do we have the most cilia in the PCT? Do we have it in the DCT? Do we have it in the rest of this? And what do we know about this? This actually isn't cilia, by the way. These are villi. These are not cilia, but that's okay. These are actually villi here. Don't worry about it. I mean, they're different, but I know what you knew, right? These are villi. And what do we know about villi? Villi and microvilli and all those things. What are they going to do? What do they change? Help with movement. Those are cilia that help with movement. Villi don't help with movement. They don't actually move, really. So Ruju and Tariq, they don't help with movement. It's all about surface area. And Sabrina, we're going to come back to that. It's all about surface area. The more villi that a cell has, the larger its surface area. Yes, no, maybe so? So you guys are darn skimpy. These are simple cuboidal epithelial cells, but in the PCT, we have a lot of surface area relative to everywhere else, especially the DCT. And you can see there are certain cells in the collecting duct that don't even have villi. Some do, some don't. That will be important. We'll come back to that. So, surface area, what is that going to help with? Everybody, you can look back at Sabrina's answer, and maybe that'll give you a clue. Just like in the small intestines, the larger the surface area, the larger the area for absorption to occur. Now, we had to learn a critical word change, is we're not talking about absorption anymore. Absorption is when we eat something in our diet, it's never been in our blood before, and we're absorbing it for the first time. Here, it's going to be called reabsorption because it was already part of our blood, then it left our blood to go into tubes, and then we're taking and sucking it back up again. Make sense? So when you guys are writing the word absorption, try to remember to write reabsorption. There is a difference between the two in the human body. But yeah, the surface area here in the PCT is much larger. So which one of these is going to have a greater role in reabsorption, the PCT or the DCT? Good. So just like the small intestines, the first part of this tube system is most important for reabsorption. Does that make sense? In fact, we're going to reabsorb well over 70 to 80% of all of the material that gets filtered. We're going to reabsorb the large majority of that, boom, right here in the PCT. Most of it gets reabsorbed right here. The DCT, just like in the digestive system with the large intestines, the primary role of the large intestines is to concentrate your waste. Well, the primary function of your DCT is to concentrate your urine. We'll come back to how it does that in just a bit. The DCT and the collecting duct worked hand in hand. Both of these, DCT and collecting duct, will concentrate your urine. You'll notice there are two major types of cells in the collecting duct. Some of them have villi and some of them do not. The ones that do not are called principal cells. You guys get to abbreviate those as P cells, which hopefully you're giggling like a little kid because they help you pee. They help concentrate your pee. They're called P cells. We'll talk more about those. And then intercalated cells. Intercalated cells are critical in sodium reabsorption. Sorry, not sodium. Potassium and hydrogens and bicarbs. That's what the intercalated cells are all about. They're about 1, potassium, 2, hydrogens, and 3, bicarbs. We'll be coming back to these intercalated cells pretty heavily by the end of the week as we're going into pH balance. Intercalated cells, potassium, hydrogens, and bicarbs. That's what they're going to help us with. Now, you guys notice something crazy weird going on with the thick segment of the loop of Henle versus the thin? What do you notice is different about the thin? What's happening to the epithelial tissue? What type of epithelial tissue do we have down here? Good, simple squamous. Simple squamous. And simple squamous cells don't have enough of a cell border for transporters. So these simple squamous cells aren't going to have your typical transporters that are moving ions and stuff like that. We're going to see that there is something else that gets transported here. So due to the change in the structure, we change its function. We'll come back to that, but I'm just making sure to point it out right now. Let's skip down to here real quick. We'll come back up to filtration. I want to show you guys that there is a slide that goes through each one of these. So for instance, what is the epithelial tissue found inside of that glomerulus? Both the parietal and the visceral layers are going to be made up of a capillary bed. So the glomerulus itself is capillary, so it's made up of simple squamous epithelial cells. The glomerular capsule, this parietal layer, is simple squamous too. It's not exactly a hard outer shell. It's not an egg shell like a typical capsule. versus the visceral layer, which is a podocyte and a basement membrane, and down below that is the fenestrations of the capillaries. So very thin, and we're going to be going into detail about that. Proximal convoluted tubule. Look at all of these microvilli, and look at all the mitochondria. Kind of tells you how much energy these are going to use. Yes, no? The more mitochondria, the more energy a cell is going to be requiring. So this is going to be very ATP dependent. Now look down at the DCT. Less microvilli and less mitochondria. So a lot less ATP driven. Why is that? We'll just make it simple as we possibly can, is that the PCT is where the large majority of reabsorption occurs. And reabsorption is going to require protein transporters, and a lot of those transporters will be ATP transporters. They'll be pumps. Make sense? DCT still has quite a few mitochondria, by the way. DCT still uses a lot of energy. The nephron loop, right? You'll notice that these are these simple squamous cells with some microvilli on them. It's not completely lacking. So there is a surface area that will help with the reabsorption of something here. And then we got P cells and intercalated cells. P cells, very few microvilli. Intercalated, more microvilli. But they're simple cuboidal cells, and simple squamous cells. So walk your way through that system. Make sure that you guys know what type of epithelial tissue is found where. And I have cross sections of all of these as well for you. I cannot emphasize enough, structure determines function, right? So if I have a tube with more microvilli, that means that there is more surface area, which means that the primary role is reabsorption. The PCT is responsible for reabsorbing nutrients from the filtrate. The DCT makes adjustments to the composition. It is concentrating the waste. Portions of the collecting duct are going to be working on osmotic gradients, and other portions of that collecting duct are going to be relying completely on hormones for their function. The thin limb of the loop of Henle versus the thick limb, each one is going to be having a different function. So keep this all in mind as we're going through, okay? Fair enough? You guys with me? Don't worry, we're going to be redundant. So if you didn't follow all that, we will see it again to some degree or the other. I will be asking you questions on it again. Okay. Let's go ahead and, yeah, let's do this. We're going to go and we're going to talk about the fact that the #1 role of your kidneys is to filter your blood. 180 liters per day. Therefore, a major part of the anatomy of the kidneys is going to be related to their blood supply. If we're filtering 180 liters of blood per day, we better understand the blood supply going to these nephrons. Yes, no? Always more to learn here. Okay, so how do I want to go about doing this? I like the idea. Let's go ahead and use, I mean, this is a pretty okay one, but I'm not here. This is a really good one. We'll come over to here. So this one's actually a flow chart too, and we're going to walk our way through this flow chart. And then we're going to write this flow chart down. Everybody, don't write anything yet. Just follow along, please. If you're paying attention, following along, you will learn this. Something you already know is that there's a big, huge artery over here called the descending aorta. More specifically, it's the abdominal aorta. Do you guys remember that from unit 2? I mean, it's a huge blood vessel that's descending all the way down from the heart. And then it's going to branch, and it's going to branch at the iliac or common iliac arteries down below. But here in the middle of the abdominal aorta, there were two very large branches, one that goes to the left kidney and then one that goes to the right kidney, and these are called renal arteries. So all the blood supply to your kidneys are being supplied through the renal artery. So that's where we're going to start our flow chart. Everything starts at the renal artery. Now, it's going to enter the kidney through that hilum or hilum. Hilum? I can't remember. Somebody even reminded me on how it should be said, but whatever. And it's going to branch. So it's going to actually diverge right as it enters. This divergence of the renal artery means that we have to change the name. So as the renal artery branches, it's going to go to just different lobes of the kidneys. You guys don't need to learn about the different lobes of the kidneys, but it's going to go to different lobes. So this is going to go in between the lobes. It's called it's called segmental. It's going to go out towards these different lobes. So it first is going to branch into a segmental artery, and that segmental artery is going to immediately diverge as well. And it's these portions of the artery that go in between the pyramids. In between each pyramid, so in between each segment. If it's going in between each segment, we call it interlobar artery, in between each one of these pyramidal lobes. So let's go to a model and just follow blood supply real quick. So it doesn't really matter which model we choose. We'll go ahead and do this one. What is this big blood vessel going into the kidneys going to be called? So that's the renal artery. And right when it branches, what's it going to become? Renal artery goes to? Good, segmental artery. Now, that segmental artery is going to branch pretty quickly thereafter. And each one of those branches is going to go up in between the pyramids. So what is that portion of that blood vessel going to be called? Interlobar. Great job. Interlobar artery right there. Now, what's going to happen next? And I want to show you guys this on this particular portion of the model. Remember, this is just one piece of the pie. So this is going to be one of these, both the renal cortex and the renal pyramid. So if this is the renal pyramid and this blood vessel right here is right on the edge of that renal pyramid, what blood vessel is this going to be? Perfect. That's the interlobar artery. It's just zoomed in. Everybody see that? So what's going to end up happening to the interlobar is it's going to make it all the way out to the cortex, past the pyramids, and it's going to make it to the cortex. And what it's going to do is it's going to branch and it's going to arc over the top of the pyramid. It's going to divide the renal pyramid from the renal cortex. So the interlobar artery is going to come up, branch, and it's going to arch over the top of that renal pyramid. And in Latin, arch, making an archway, is arcuate. So A-R-C-U-A-T-E. So we have the arcuate artery. It arches over. It's the arcuate artery. So if I put a pin here, anywhere between the cortex, the renal cortex and the renal pyramid, those are all arcuate arteries. Now the arcuate artery is going to branch. And I want you guys to look at this portion of the model first, because I think it's pretty clear over here if you just stop looking at all the words that are in your textbook and stuff like that. The arcuate artery, do you guys notice how it's got these branches that are radiating out throughout the cortex, moving out towards that capsule? They actually make it all the way to the capsule, and they're going to be feeding the edge of that capsule. Yes, no, maybe so? Now, parts of your book and parts of the lab objectives, they call these the interlobular. And I just don't like that term because it's too close to interlobar. My brain doesn't hold that very well. I don't know why, it never has. So there's actually another name for these that I suggest that you guys learn. And I like it because it actually helps with its function. It's called the cortical radiate arteries. I just wrote it down in chat for y'all. Cortical radiate, because it radiates out throughout the cortex. It radiates from the arcuate artery, and it's radiating out away from that. I personally like that term better, but if you want to use interlobular on the lab practical, just make sure you guys don't get mixed up, interlobar and interlobular. But yeah, if you look at these two models, remember this is just a zoom in, what you'll notice is that we have the arcuate artery here, and it branches and radiates out towards the cortex. So this, that my pointer is over right now, this, that my pointer is over right now, these are called the cortical radiate arteries. The cortical radiate arteries are, you got to remember, these are diverging. They're getting smaller and smaller and smaller as they go. Cortical radiate arteries are pretty small. They're still arteries, but they're pretty small. And then when they make their final branch, when the cortical radiate kind of looks like a fern or a plant, you know, with branches off of that plant, these, and therefore they, you know, kind of lose that tunica externa, and we get that smooth muscle band around it. I just glitched. Everybody hear me okay? Okay, cool. So these right here are going to be the afferent arterioles. So the branches from the cortical radiate, those are the afferent arterioles. Cool beans? Let's walk our way back. We can't just talk about blood supply to, we have to talk about blood supply away. What you'll notice is that these blood vessels here are red. The afferent arteriole is red. The efferent arteriole is red. Why? Because gas exchange does not occur in the glomerulus. This is where filtration occurs, not gas exchange. Big difference. So what's going to happen is the afferent goes into the glomerulus, the efferent comes out, and now we have our typical continuous capillary bed. Okay, do you guys see this capillary bed right here? We got all these branches. It's going to go from red to blue. So this is where gas exchange is going to occur. Cool. Now, let me show you guys on some other pictures what's actually happening here, because this model doesn't do a great job representing what's really happening. What's really happening is that we have this capillary bed that wraps and weaves itself through the tubes. All these purple lines here are part of this capillary bed, and they're surrounding and wrapping themselves around the PCT and the DCT. Everything that's inside of the pyramids is wrapped up in a big capillary bed. These are called peritubular capillaries. They're always going to be associated with the convoluted tubules. They're going to be associated with the renal cortex. Peri meaning surrounding or around, tubular meaning around the tubules. Peritubular capillaries. Totally tubular, dude. But those aren't the only capillary beds. The other capillary beds, you'll notice, look different. There are capillary beds that wrap around the loop of Henle. And they got these big, long, straight lines. Very different than your typical capillary bed. And straight, if you guys didn't learn straight in Latin and AMP1, it's recta. Totally tubular? No, that's from Teenage Mutant Ninja Turtles, dude. Yeah, old school. But anyways, straight in Latin is recta. That's right. Turtle Bauer. First movie I ever saw in a movie theater by myself. I rode my own little BMX bike to the movie theater to watch the original Teenage Mutant Ninja Turtles. I felt like such a big kid. Back in whatever that was, 84, 85. Crazy, long time ago. All right. Vasa recta, vast, straight. That's what that means. Vasa, vast, big. Recta, straight. These are big, straight capillaries. That sounds about right. 7, 8, something like that. So what do you guys notice about the peritubular capillaries? Renal cortex. What do you guys notice about the vasa recta? Renal pyramid. So now let's go back to the model over here, and maybe this will make more sense. There is a capillary bed in the renal cortex, and there's a capillary bed in the renal pyramid. What is this capillary bed in the renal pyramid called down here? If I were to put a pin down here, what is this blood vessel structure? Awesome, awesome. Keep going, guys. Say it out loud, yell it out loud, whatever you got to do. That's the vasa recta. And the reason why it's a little misleading is because people are looking for the loop of Henle. But of course, this is a model, so they're dissecting the capillary bed away from those loops. So this vasa recta would actually be wrapped around this loop of Henle. It would be wrapped around this loop of Henle. Yes, no, maybe so? Whereas this peritubular capillary bed would actually be wrapped around everything. It's around all of these tubes. It's not just found in this location. So please don't think that. I've had lots of students in the past that, yeah, that's what they're thinking. And there's a number of different models that I have to represent this, but capillary beds around the tubules, peritubular, capillary bed around the loop of Henle. Lots of different blood vessels. There's the arcuate, cortical radiate, going out to the glomeruli. So, all right. So, let's put this all together. Everything we've done so far, let's put it together into something that you can study. I think that's always the hope. Let's start with the blood supply. We know that it all comes in via the renal arteries. So renal artery. Left and right renal artery, let's just deal with one kidney. From the renal artery, it enters the kidney and divides into a segmental artery. Segmentals are going to divide, and each division of the segmental artery is going to go in between the renal pyramids, in between each individual lobe. So we call it interlobar arteries. The interlobar artery is going to pass all the way through that renal column up into the renal cortex, and it's going to divide the renal cortex from the renal pyramid. It's going to arch over the top of all of that. So we call it the arcuate. And then the arcuate artery is going to branch, multiple branches that radiate out through that cortex. Lots of cortical radiate arteries just branching out throughout the entire renal cortex. But ultimately, each cortical radiate artery is going to have multiple branches itself. Those branches are the smallest of the branches, surrounded by circular bands of smooth muscle. I'll say that over and over and over. But these are going to be that afferent arteriole. Now, the afferent arteriole, I hope you guys recall, feeds into the glomerulus. Yes, no, maybe so? You're going to get, 1,000 liters of blood that go into your kidneys every day. And of that, 180 liters are going to be filtered. Okay, so as it gets filtered out of that glomerulus, it's going to go into that capsular space of the Bowman's capsule. We'll come back to that part of the flow chart in just a second. But whatever doesn't get filtered is going to leave the Bowman's capsule. It's going to exit. And what's it exiting that glomerulus via? The efferent arteriole. The efferent arteriole can go two different places. One, it could branch and go all around the capillary bed in the renal cortex. Well, that's going to be the peritubular capillaries. Alternatively, it can actually dive down into that renal pyramid, and it's going to surround that nephron loop. Big, long, straight branches of capillary. Very unusual. Yes, whatever is not 80% of what goes through your kidneys is not filtered. Only 20% of what goes through your kidneys gets filtered. So 80% goes boom, right back out the efferent into these capillary beds. These two capillary beds can also kind of communicate with one another, but we won't really deal with that. As gas exchange occurs, of course, now our blood starts to turn more blue, because now it is CO2 rich, because we got gas exchange. Good news. I got great news for you. We just follow it all the way down. It doesn't move at all, Melissa. We can't move 100%. If 100% of the blood going into your kidneys was filtered, you'd basically not have blood. It'd be always in the tubule system. What was it again? What do you mean? Help me out. What was what again, Melissa?
I guess what I was talking about, I'm just trying to relate it back to the hoarder's example. So you were saying, so a lot of it goes back.
Yeah. Yeah, that's all going to occur out here. So my example, the desk example was a little bit more accurate in numbers. So I said, you just take the dirtiest desk in the world, you put your arm and you sweep 20% of that into the garbage can. The garbage can is the Bowman's capsule. So 20% of your blood's going to get filtered right here. 20%. Does that make sense? Does that help? So this is 20% of your blood's going to get filtered. So the other 80% is now going to just pass through here. This is all in the garbage can. So now we got to go pick it back up. So that's what the rest of that tubule system is. It's the, whatever, the hoarder dude going back and saying, no, I ain't throwing that away. Nope, not throwing that away. No, sir, no how. Right? It's this 20%. That's 180 liters is going into the Bowman's capsule. But that still means over 800 liters is going back out into the efferent arteriole. Tariq, you have a question?
Yes, Professor. So I'm trying to understand this concept. You said 20% gets filtered. So let's say if you take a bucket, right, of clean water, and we put a drop of black ink in it, and it will take time that that black ink would get saturated around. So if you're only taking the 20% at the bottom of the bucket every time, how the system will recognize that out of every time the 100% we're taking 20% out of of the blood.
Great question. And we will come back to that. I mean, I'm not leaving that behind. That's all part of the physiology. It's through something called GFR, glomerular filtration rate. We actually have cells that are responsible for monitoring the osmotic potential of your filtrate. We're coming back to all of that. We're going to get different cytokines and hormones that are going to be getting released. We're going to get nervous systems that are going to get triggered, intrinsic and extrinsic responses. You guys know the drill. All of that is going to be regulating how much gets filtered. This is the most critical value right there. That 20%, crazy, crazy critical. It's called glomerular filtration rate. If your GFR is too high, bad things happen. If your filtration, your GFR gets too low, bad things happen. So the body's going to have a lot of homeostatic variables controlling that. Helpful? Does everybody follow that? Not just Tariq who asked that question? We are going to be controlling that. But before we can do that, we got to learn the basic anatomy of the system, of course. So now blood is leaving, because another important thing is if we're reabsorb. Oh, I like this part. Y'all, we're filtering 20% from the blood. We don't pee this 20% out. Can't do that. So we're going to reabsorb the large majority of that. Where's it getting reabsorbed into? We're picking through that garbage can and we're putting it back where? Where did it come from? It came from the blood. So where does it have to go back into? It's got to go back into the blood. Okay, so from this tubular system, it's going to be going back into these capillary beds. Does that make sense? Cool. Anyways, the system back's pretty simple. Vasa recta, peritubular capillaries, both of those can go into a cortical radiate vein, or they can actually bypass the cortical radiate vein and go directly into the arcuate vein. The arcuate vein goes into the interlobar vein, passing back down between the lobes. There's only one change to the story here, is that there's no such thing as a segmental vein. That doesn't exist. Okay, so segmental artery does not have a mirror image vein. All of the interlobar veins are going to immediately go back into the renal vein. The renal vein, of course, is going to go into the inferior vena cava. So it's going to feed, boom, right back into the heart from there. So we might as well actually, since we did it, no, you guys know, right? This is coming from the descending aorta, the abdominal aorta to be more specific. Just don't want to take up too much space. Does everybody feel pretty comfortable with the flow chart for blood flow, blood supply? Where do we regulate blood going into and out of the glomerulus? Arterioles. Arterioles, smooth muscle that we can control, nervous system control, adrenergic receptors. You guys see the importance in learning these words, arterioles, and having a image in your head of what an arteriole is. Yeah, definitely looks like heart blood flow. It's just, yep, you got it. Arterioles, though, we gotta kind of think when you hear arteriole, we know the sympathetic nervous system is going to have an impact, adrenergic receptors, so forth, so on. So let's tie this all in. Bowman's capsule. From the Bowman's capsule, now we're going to head out into the PCT. From the PCT, it's going to descend into the loop of Henle. There's, of course, a descending limb and an ascending limb, both thick and thin, but that's going to go back up into the DCT. The DCT all feeds into the collecting tubules, which feeds into the collecting duct. The collecting duct is going to run all the way down through that renal pyramid until it makes it to the renal papilla. From the renal papilla, it's going to go, kind of running out of room here, sorry, minor calyx to the major calyx, to the renal pelvis. Renal pelvis feeds into the ureter, ureter to the urinary bladder. I'm totally out of room here. And from the urinary bladder out the urethra. Is everybody able to follow along here? Tried to be redundant, tried to show you get doing this, you know. Nah, you'll be fine. And yes, by the way, PRCC, papillary renal cell carcinoma. Yeah, it's all happening down there in the papillary. In the papilla. I love it when people make connections, right? Melissa, you definitely, you know, your heart and soul is in cancer and learning more about it. I appreciate the heck out of that. Good flow chart for you guys to be learning. Let's take and remind ourselves that this is your lab objectives. I'm not just doing this for lecture. I'm not just doing this for poops and giggles. This is all part of your lab as well. So if we come up here to renal anatomy lab and we look at what we have available, we have drag and drop activities, we got urogenital models, we got urinary system lab models, a lot of blank pictures to work off of, and we have there are our lab objectives right there. Identify the structural components of the urinary system on anatomical models. Renal cortex, renal medulla, renal capsule, the hilus, renal pyramids, renal papilla, renal calyces, major and minor, renal pelvis, renal sinus, ureter, urinary bladder. We haven't discussed the different parts of the urinary bladder and the urethra. Does everybody feel like they'd be able to identify each one of those after a little bit of practice based on what we've already done? Flow chart for blood supply, renal arteries, segmental arteries, interlobar arteries, arcuate arteries, interlobular, same thing as cortical radiate. Look, it's a flow chart, y'all. Afferent arterioles into the glomeruli, out of the efferent arterioles to the peritubular and vasa recta, interlobular, cortical radiate, right? Veins, arcuate, interlobar, renal, no segmental. Does everybody feel like with a little bit of work, they'll be able to identify each one of those blood vessels? I didn't get as many yeses there. I hope the answer is yes. We good, powered through that together. We'll come back to what's going on with microscopes in just a second. Identify and follow instructions on nephron models. Nephron models are these models and these models. Okay, so we're zooming in onto a nephron. We can also look at it from the outer cortex here, or from the outer cortex, from the bigger kidney model. But the nephron, of course, is what we've been focusing heavily on. And then this is just the glomerulus model. So if we go back and say nephron models, the renal corpuscle, which is the Bowman's capsule, plus the glomerulus together, we need to be able to identify the Bowman's capsule, both the parietal layer and the visceral layer. On that visceral layer, we should be able to find podocytes and filtration slits. As we follow those nephron tubes, it goes proximal convoluted tubule to the nephron loop, thick and thin, descending and ascending, and comes back up into the renal cortex through that distal convoluted tubule, all into that collecting duct. Should be able to also find glomeruli, afferent, and efferent arterioles on one of these models. And we'll come back to the juxtaglomerular apparatus in just a second. So everybody feel like they'd be able to identify everything on here except for the juxtaglomerular apparatus, because I have not followed that yet. Sweet. Okay, cool. I hope you guys can see that lab and lecture have definitely coincided. Now, I've also tried to prepare you guys for histology so far. The histo isn't too crazy. It is definitely the hardest part of this lab. Microscopic slides, renal capsule, renal cortex, renal medulla, glomeruli, glomerular capsule, PCT, and DCT. But let's see if we prepared you well enough to be able to do some of these. Yeah, it's because I didn't go through it with you guys specifically. I gave you guys access to everything to do it on your own. But if you're not going through it heavily, then you're always going to get thrown off. Just histo's not easy. Just the reality of it. It takes a lot of practice. Urinary system. Sorry. Okay, urinary system. So what we want to focus on primarily is going to be the urinary bladder and the kidney itself. So with the kidney itself, let's just look at the, the big picture here. What I want you guys to see is there's some color differences. For instance, deep down in here, it's this light pink. Out here is dark purple pink. I know you guys are like pink, purple, histo is just a bunch of different colors of purple. But I've tried to teach you how to do these landmarks already. So as I zoom in, what are you guys noticing? What's going on right here? Little circles. So what might these be based on everything that you've learned so far? What might be a little circular structure found throughout the kidney? I love that you wrote nephron, but that's not exactly right. That's not, remember, the nephron's the whole thing. The nephron is everything you see here. What I'm asking you guys is about a circle. Bowman's capsule, glomeruli. What were the name of those two things together? What's the Bowman's capsule plus the glomerulus together called? The glomerular corpuscle or the renal corpuscle. Okay? The capsule is just that outer shell, but the corpuscle is everything together. The corpuscle, the renal corpuscle, is both the glomerulus and the glomerular capsule combined. Is that clear? Easy to miss from earlier's lecture. I get it. Easier to miss. So if we go back here and we see one of these, we know that this whole unit is a corpuscle. Okie dokie? Corpuscle, corpuscle. So if I just want to zoom in on this a little bit, I can do that, and I'll come back to my point. But as I zoom in, you'll notice that there is something here in the middle. There's a white space, and then there's an outer line. Yes, capsule is the hard candy shell. You got it. Glomerulus chocolate. Yummy. So good. I was at a coffee shop the other morning, and it said, coffee, chocolate, and mint. Some things are better rich. I laughed out loud. I was like, probably not wrong. All right, so that's what we got going on here. We got a capsule. This is that capsular space, that open white space right there. So everything outside, you guys see this tiny little layer of squamous cells? The nuclei are squashed even right there. Those long, flat nuclei tell us that this is a squamous cell. That is the Bowman's capsule. And everything in between here, all of this is going to be the glomerulus. This one is great because this one even shows portions of the afferent arteriole, which is pretty darn cool, very unique, not easy to see. You will not need to identify that on a lab practical, but still pretty cool. So I got a glomerulus, I got a capsular space, and I got a Bowman's capsule. So all of that is a corpuscle. So what do we know? 100% of what is occurring here? Good, Andy. Good correlation to what we learned earlier. 100% of filtration is occurring here. And so what region of our kidneys does 100% of filtration occur in? Good. See the association. I tried to get you guys to have a memory for this, and it seems like it worked. Awesome. The renal cortex. So if I find on histology, I can see these little round circles. I know that they are corpuscles, renal corpuscles. Therefore, what region of the kidneys do I know I'm in? No ifs and or buts. Don't mess around with it. That's the renal cortex. And then if I have a portion of the kidneys that does not have a renal cortex, what am I dealing with here? That's all renal medulla. That's all renal medulla. And everything that you guys see here, all these lines that you guys see here, those are all collecting ducts. Those aren't actually the loops of Henle. Loops of Henle you're not going to be able to see very well. Loops of Henle are going to be part of just this upper region here. These are all parts of the collecting duct that are going down to the, what do I got down here? You can see it actually really well. You do not need to identify this on histo. I'm just showing you. Good. This is a papilla. This is a renal papilla, and it's going into a minor calyx. Cool. You guys don't need to be able to find renal papilla or a minor calyx, but it does exist. Awesome. We can do that with a number of these, all of these different kidneys with the different staining. If I have, come on, focus. If I have these little round circles, what do I know I am? What am I if I'm this little round circle? No other organ in the human body looks anything like this, by the way. It's the only organ in the human body that has all these little circles like this. So these are all part of the glomerular corpuscle or the renal corpuscle. There's a Bowman's capsule in there and a glomerulus in there. Awesome. All right, so the hardest thing to learn here, again, I've tried to prepare you for it, is you will need to be able to identify the proximal convoluted tubule versus the distal convoluted tubule. So now is when we're going to look at all the rest of these tubes. These are cross sections. So everything you guys see here, all of this space, all of this space, all of that, those are all, if it's not part of the corpuscle, then it's a tube. It's a tubule. Yes, no, maybe so? You can go PCT. And you can go DCT. Now, I'm going to just zoom out a little bit. There we go. Maybe a little bit too much. But I want you guys to see that there is a couple of notable differences between the two. The staining on the two is different, first and foremost. So if I zoom in here, you guys notice that this tube right here, the individual, come on, the individual cells are bigger. The spacing between the cells are bigger. There's less cells surrounding this tube. Yes, no, maybe so? You can see it over here as well. There's less spacing. The cells are more spread out. That's one. Whereas over here, these tubes, you'll notice that the cells are very concentrated and they're kind of a dark purple. Yes? That's one way to be able to tell the difference between a proximal convoluted tubule and a distal convoluted tubule. You just look at the cells. The other way, and this is the way that I am going to make sure that I, represent my picture on your guys' lab practical, has to do with function. Yeah, and the size too, because if you go back to the lecture and you go here, which one had a bigger cell? Which one has more spaced out? Which one is thicker? The PCT. And which one has a greater surface area? PCT. So when we go back here again, we see bigger cells, more spread out, and look at the inside of this tube. Look at the inside of this tube. You guys see, it looks like there's something shoved into this tube. But that's not something shoved into the tube. Those are all microvilli. That's not something shoved into the tube. They're microvilli. And now look at these ones here. The tube looks open. The tube looks almost clear on the inside, doesn't it? It's smoother. So dark purple cells, that's one way to identify it. And then the spacing in that tube, the opening of that tube, the blurriness of that tube, that's another way. So let's do this. You guys can use abbreviation here. What specific type of tube do we have right here that I'm circling with my mouse? And what specific type of tube do we have right here? What about over here? What's this? What's this one right here? What's this one over here? You guys feel pretty comfortable with that? Just about practice now. Practice. Practice. You could go almost anywhere on this roadmap here. Check out this section right here. It's almost all PCT, but there are some DCTs in there. There's one right there. There's one right there. So you can see them in there. They do exist. DCTs here. Yeah, just practice. Practice. Cool. Let's finally go down to the bladder. Bladder's pretty easy, but I get a lot of students that miss this every semester, and I don't know what I do wrong. So try to pay attention here. The urinary bladder has a very unique epithelial tissue. You only find it inside of the urinary bladder. Yeah, bacon. You only find it inside the urinary bladder in the upper 1/3 of the urethra. Those are the only two locations in the entire body to find this type of epithelium, and it's called transitional. Okay, this is called transitional epithelium. It's supposed to be one that you learned in A&P1, and students just don't remember it because it's so rare, I guess. So maybe it's just not emphasized enough. Transitional is called transitional because it transitions. They are highly stretchable and they are highly recoilable. So what sort of, I don't know if recoilable is a word, but I use it anyway, so deal with it. So if it can stretch and it can recoil, what kind of tissues do we know is going to be found throughout all of this entire urinary bladder? Elastin. We know there's lots of elastin fibers. Great, awesome. Now, transitional epithelium can either look relaxed, or because it's a bladder, it can look stretched out. And when it looks stretched out, you'll notice that the epithelium looks different. You guys see how it's more flattened and that the surface is like almost perfectly smooth? So it can transition between this state and the relaxed state. Now, for your lab practical, I promise I will use some form of relaxed. I just think it's easier to see. And the relaxed transitional epithelium always looks like, I call it bubble gum. Okay, it looks like there is popped bubbles or bubble bath or something like that. You got bubbles, bubbles, bubbles. My bubbles. Yes, no, maybe so. This very top cell, this big round cell that's on the top, this is the stretchy cell, and it's called an umbrella cell. Umbrella, Ella. You won't forget it if you just sing Rihanna every single time. Got umbrella cells. These guys are crazy dynamic. They can change their shape real easily. Okay, the surface of transitional epithelium is coated in umbrella cells. These umbrella cells have lots of baroreceptors, and they are going to be monitoring stretch and strain, and there's always strain and stretch on the walls of the bladder too. What else do you guys need to be able to identify? I think it's really just, I guess, okay, mucosal layer, which is the transitional epithelium, and you need to be able to find the muscularis layer, but that shouldn't be too hard, right? Because we're dealing with the bladder, so the large majority of the tissue here is muscularis externa. It's just one gigantic circular muscle, basically. Inside layer of bladder. I guess I'm not understanding your question. The innermost layer of bladder is always going to be, the innermost layer of all organs is always going to be epithelial cells. And the particular epithelium found inside the bladder is transitional. You're welcome, bub. Where this comes into play as well, did anybody ever heard of TCCs? Oops. Melissa, maybe you. They're pretty rare, but they're called transitional cell carcinoma, transitional cell cancer. By the way, if you ever see the word carcinoma, in case you guys haven't learned this before, carcinoma is always epithelial cancer. So all the carcinomas you've ever learned about are epithelial tissue cancers, which means it's epithelial cells that are now, immortal, learn how to survive. But TCCs are really dangerous. People don't realize how dangerous they are. They are extremely aggressive, and they metastasize. They get into your bloodstream and spread throughout your body very easily. So when people say they have bladder cancer, potentially if the bladder cancer is a TCC, a lot of people will be like, I'm good, it's just bladder, man, whatever. I'll just, have a bag. I'm good. But bladder cancer, if it's in your transitional cells, can be very, very dangerous. So I just want to kind of get that out there so people, cancer doesn't kill you because it's in a primary organ killing you. It kills you because it metastasizes and spreads somewhere, and then it destroys those organs. And TCCs metastasize very rapidly to the brain. So a little bit morbid, I know, but I definitely want just you guys to be, educated in medicine in slightly different ways. Just always logically think through things. Yep. Irradiating the bladder is pretty brutal too. Okay, cool. So let's see. What have we gone through? We've gone through almost everything. I think there's only one more thing. I guess in the urinary bladder, by the way, they are covered in urinary ruga, but you guys already know that. What are ruga? Where did we see ruga before? Rugae, plural. Yeah, we saw them in the stomach. You're going to see them in the bladder. So yeah, it's basically if we have any models of the bladder, that bladder is opened up. Here we go. So we got models of the bladder, and that bladder has these big folds in it. If I put a pin in that fold, it's going to be urinary ruga. Not renal. Good try though. I like that. But it's inside the bladder, not inside the kidney. So we call it urinary. Okay? That's the male repro system. The female repro system is somewhere. Gotta have one of those too. There's some more ruga there and just a in that system there. Yeah, so there's the female repro, not very blown up, but definitely have it as well. Please, y'all, this is not the uterus. I put a pin here. Don't call this the uterus. I got a lot of students every semester that do that. Follow would you want to, I know having a baby out of either one of these canals is no fun, but I think having a baby out of that canal versus that canal might be, I don't know, slightly less painful. Maybe not a whole lot less painful, but what else? Okay, we'll be dealing with urogenital diaphragms and all the different sections of the urethra. We'll be dealing with that in the reproductive system. So we are good except for one thing. And in order to do that one thing, I want to remind you guys that this picture is linear. This is not the real deal. The real deal is here, for instance, or here. So let's use this diagram. I want you guys to follow this tubule system with me. Redundancy is the key to learning. So if we take our glomerulus, we filter from the glomerulus into the Bowman's capsule. We enter the PCT, at proximal convoluted tubule. Large majority of the reabsorption of the system occurs here. We'll be drawing that out more. It's going to descend down into the descending limb of the loop of Henle, both thin and thick, coming back up the thin to thick descending limbs of the loop of Henle into the DCT. But there's something unique about the DCT. Every single DCT twists, winds, does what it do, but I want you guys to notice something about the DCT, is that it comes right back to the glomerulus from whence it came. I love saying that phrase. I don't know why. Maybe because I'm a big-time nerd. From whence it came. I don't get to say that very often, so there you go. The DCT always comes back from whence it came. And it's going to split right in between the afferent and efferent arterioles. So again, let me show you this on any other model here. We follow this. We got a PCT, does its thing. Loop of Henle does its thing. It sends back up. DCT is twisty, windy, but boom, look. It comes back and it splits the afferent and efferent arteriole. It comes back into contact with the glomerulus from whence it came. And then finally, it's going to make its way over to that collecting duct. Let's do another one. PCT, twisty, windy, loop of Henle, descending, ascending, twisty, 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 twisty. Look, it's not quite as robust in this picture here. It shows it better here. So if we twist it and wind it away, there's that DCT coming right back up in between the afferent and efferent arterioles before going to the collecting duct. Do I need to do this again, or is this making sense? Every single DCT is going to go back near that afferent and efferent arteriole from whence it came. You can watch the recording. It's an important little fact, because what's going to end up happening is that this area where the DCT comes back into play is a very important area physiologically speaking. So you may or may not have noticed this tube that's bifurcating the afferent and efferent arteriole. It's been in every single picture that we've looked at so far. It's here, it's here, and you guys have probably just been ignoring it because I haven't been mentioning it. You're like probably going, please, please, dear baby Jesus, don't have him. Don't have him. No, don't talk about it. Maybe if he doesn't talk about it, it won't exist. It exists. This is the JGA. This is the juxtaglomerular apparatus. The juxtaglomerular apparatus. In this model, it's showing in this lime green. In this model, it's showing right up here. bifurcating the afferent and efferent arterioles. JGA is what you guys can call it on a lecture exam. Juxtaglomerular apparatus is what you'd have to call it on a lab practical. Okay? Juxta, meaning next to, in close proximity to. Juxta actually means overlying something else. Juxtaglomerular, it's an apparatus that comes right next to the glomerulus. Juxta. It's an apparatus because it's comprised of many different things. For instance, there's a group of cells that are a part of that distal convoluted tubule called macula densa cells. So you'll notice that there's a region of this DCT where we have normal simple cuboidal cells, but then there's a cluster of cells that turn columnar. Yes, no, maybe so? These are the macula densa cells. The other cluster of cells are those extra ones that Ruju noticed earlier in lecture. They are simple cuboidal cells that surround the afferent arteriole, not the efferent. And these are the granular cells. So we got macula densa cells, and we have granular cells. And it's the combination of these two cells, the macula densa cells of the DCT and the granular cells of the afferent arteriole, that's what makes up the juxtaglomerular apparatus. So draw this into our diagram. We'll go ahead and use this diagram for that. So this is just the first one that I drew. You guys can use even this one, right? You could do the same thing there, just a little bit more blown up here. So if we draw this in, I'm just going to draw, it's kind of oval, whatever. It doesn't really matter which shape you draw this. So this is going to be the DCT. Certain portions of this DCT will just be normal cuboidal cells. But now where it comes back into contact, especially around that afferent arteriole right there, it turns columnar. So we're going to start making these things a little bit more columnar. We'll just make it as obvious as we can. Clear. Those are the macula densa. Macula densa are specialized osmoreceptors. So they are monitoring the osmolarity of the filtrate. So they are monitoring, they have receptors for salts, for osmolarity. Osmoreceptors. Yep, they're constantly monitoring that party. Bouncers, I love it. I never really thought about that. Yeah, they're the bouncers, kind of. They don't actually do the bouncing though. They're going to communicate to somebody else. So these are the osmoreceptors, all right? Macula densa cells have osmoreceptors, and they are monitoring the filtrate. So there's going to be fluid inside of this DCT. This is all part of that DCT. They're monitoring the fluid inside, and they're going to be monitoring the amount of salt there is. And they're going to be in communication with a cluster of cells that are out here. These cluster of cells actually that they kind of actually are all on the inside, and the glomerulus kind of wraps itself around these cells. Didn't do a very good job at drawing this, I'm sorry. Because that's not part of the Bowman's capsule. There's a cluster of cells in and of themselves. Those cells in there are called mesangial cells. First time you've heard that term. In here are mesangial cells. You do not need to know mesangial cells for lab, but you will need to know mesangial cells for lecture. Mesangial cells are basically little cells that have contractile properties. And the glomerulus kind of wraps itself, grows itself around those mesangial cells. The last cluster of cells you guys need to know about are the ones that surround the afferent arteriole. Okay, these are some simple cuboidal cells. These ones are called granular cells. Everybody caught up in their labeling so far? Let me know when you're ready, Melissa. Ha! No, are you kidding? You're such a dork. Yeah. No, I just like colors, and I've always been a fan. My professor, my physiology professor, again, I'll never forget Dr. Phil Sanger. He was my mentor. He changed my life. He taught me how to study. He taught me how to be a student. And used to play racquetball with him. Yeah, I ended up working for him in his labs after work. Afterwards, he was good friends with Gary Larson, so he was working on a repro textbook because he's like the repro master of the world, repro physiology master of the world. So he was working on a textbook with Gary Larson. Super cool. He did everything on a blackboard with colored chalk. That is it. My entire class. And ever since then, I've been studying, trying to use as many different colors as possible. It helps my brain break apart each idea, each concept. It just works for me. Oh, nice. Yeah, I haven't played in a long time, but used to play lots of tournaments once upon a time, many, many years ago. Melissa, are you ready to go? You got caught up? Anybody? Everybody? Melissa, are you ready to go? Are you caught up? Okay, cool. Tariq, you're ready. Melissa, are you there? Hello? All right, I'm going to assume everybody is caught up on this. I slowed down here because of how important this next part is. These granular cells aren't just a name, okay? They are a very important cell that maybe you recognize from earlier this semester. I did mention them once upon a time. These granular cells are a very important cell for helping with blood pressure. Blood pressure is going to be a big part of this story. Blood pressure is going to help determine how much filtration occurs every single day. So if a lot of filtration occurs or a little bit of filtration occurs, that can be a measure of your blood pressure. As such, a major hormone that helps to control blood pressure is secreted from your kidneys. Which hormone is this? What hormone am I talking about? This is renin. Granular cells are the cells surrounding the afferent arteriole. These are the ones that secrete renin. Guys gotta know renin. Got stuff. So if blood pressure is too low, what hormone will be secreted? If blood volume drops, if blood pressure drops, what hormone is going to be secreted? Renin, motherfucking renin. It's going. And it's going to end up fixing, hopefully, that problem, unless you have a disorder or a disease. And now renin gets all wonky. We're going to learn how it does this, but it's all going to come back to this juxtaglomerular apparatus because of how filtration works and how it relies on blood pressure. So when blood pressure drops, filtration is going to start to drop, and if filtration drops, then the salt concentration is going to drop. And as that's, or not drop, sorry, the salt concentration is going to change. It does drop, but I'm going to explain to you why it does that. And as that salt concentration changes, the macula densa is going to be monitoring for that salt. And those macula densa cells now can secrete cytokines that are going to be affecting that afferent arteriole and the granular cells that surround it. Convoluted, but super cool. Let's see, what do we want to do? Here's some GFR regulation. How? Just listen to me whine right now. How? That's what I'm doing when I grade exams and I don't see the how. And I write down how on your exam. That's me going, but how does it work? Nap. Just double checking to see what we've gone through real quick, because we've basically gone through everything, either directly or indirectly. For instance, urinary bladder. What type of epithelial tissue do we know is on the surface of the mucosal layer in your bladder? Help me help you? Just quizzing you now. What type of epithelium? What type of epithelium? Not what type of cells, Tariq. So transitional epithelium. This is one that I think probably is the most missed question in the history of my lab practical. I don't know why. I don't know. So make sure you study it. Hint, hint. I mean, it's not even a hint. Just study it. You should know it. If you find umbrella cells, we know that we're in transitional epithelium. They're the only cells in the human body. Sorry, they're the only epithelium in the human body that has umbrella cells. No other epithelium in the human body has it, so it should be obvious. Detrusor muscle, that's their muscularis layer. This is not inside the peritoneum, so the connective tissue that surrounds it's called adventitia. You know, the normal stuff you guys have been learning about. Different urethras, female versus male, but both of them have an internal urethral sphincter right at the base of that urinary bladder. And both have external urethral sphincters. Females is right here in the urogenital diaphragm, sweet. Males, it's also within the genital diaphragm. So this is the pelvic floor for people that are like, what the hell is a urogenital diaphragm? That's the pelvic floor. And that's why when a baby comes through here, not through this hole, but right behind it, it can cause damage to this external urethral sphincter. So don't be jumping on trampolines too much immediately after having a baby. Males, we have a little bit more going on in terms of space, but this is also a problem because if males hold their bladder too long, it actually causes, because the internal urethral sphincter, that's that reflexive relaxation. So urine is going to build up inside that prostate gland. And then we have the external urethral sphincter here that's contracted. So as we need to pee, more and more urine is going to be bouncing around in here, increasing the pressure, and it can actually cause inflammation of the prostate gland if you try to hold your pee too long. So anyways, a couple cool things, but we'll deal more about the female and male urethra for the reproductive system. Yeah, let's deal with filtration tomorrow. Let's go ahead and get out of here a little bit early. I'm pretty proud of what you guys were able to accomplish today. I think do you guys feel comfortable with the anatomy? Veronica's like, peace out, gone. You guys feel comfortable with the anatomy? I mean, if we would have done this day one this semester, you would have just quit. But now we can go through it at a much faster pace. You guys are following it a lot faster, I hope. And at the very least, you know that you need to go sit down and study it on your own a little bit, but that once you do, you have a route to be able to study this material and hopefully retain it a little bit better. I'm pretty proud of you guys and your answers, so be proud of yourselves. If there are any questions, let me know by text. Tariq, I got your texts. Just we'll go over to office hours. And cool. Everybody else, text me if you need me. Just because I'm on vacation doesn't mean I'm not handy. So let me know. Laters, y'alls.